All right, let's come back. So next topic, we are going to talk about quality recalibration. So you can see that from raw data to alignment and doing alignment to the BAM file, which is aligned reads, and then to the variation calculation. So what we are trying to get here is for the raw data part. And remember, there's 3 levels of qualities: the base quality, the mapping quality, the variant quality. Okay, we are trying to say that the raw quality that the sequencer reported, sometimes we just take it for granted, right? If we got an Illumina sequencer, it tells us this base is a G and the p-value is 0.0001. That's pretty good, right? And the quality is a 50 or something like that. It's really, really good. But do we really believe it? Okay? I didn't challenge it until this paper published, right? And let's review a little bit what quality really means. We are talking about here is the base quality. So you see this is the quality value, and the general we are using this kind of Fred type of scale. And long story short, this p-value is the smaller the p-value is, the better quality this particular read is, and therefore this QSeq will be better. Okay? And the base quality reported by the sequencer part, are they really accurate? So without a positive control, it's really hard to evaluate whether they are accurate or not. And the positive control meaning you know exactly what you are sequencing, and which it will be never the case, right? But without a positive control, it's hard to evaluate. And this is a paper published in Nature Genetics a couple of months ago. So you can see that this is, I'm sorry, this is a little small. And this is 4 different platforms. And this is the Illumina GA2, which is an Illumina earlier platform. This is a 454. This is solid, and Illumina HiSeq 2000. Okay? So if you look at this, is X here is reported quality. Okay, if there's a 0, that means it's really bad, and 40 is meaning the quality for that read is really, really good. And the Y here is so-called empirical quality. The so-called empirical quality meaning that it's estimated quality based on the real data, not based on their reported data. Okay, so focusing on the pink dots here. So each dot represents all the base with this particular reported quality. So you can see this, for example, this one, that represents all the base they reported as 30, quality as 30, which means the p-value is 0.001. It's pretty good, right? But when you look at here, if they reported this is a 30, the empirical quality here is about 25. Illumina lied. Okay? All right. So the theoretical value should be very close to this diagnosis, right? For this close to 45 degree, but Illumina really lied. And 454 is doing okay, and solid is doing okay. For Illumina HiSeq 2000, and same issue showed up. And how they really calculate the empirical quality, so it's worth looking at a little bit. So X in this figure is actually the reported quality, and Y is the empirical quality, which is a mismatch at the loci without known variance. Okay? So if for the region that it's not in dbSNP, or so we know that there's no reported variance there. And if there is a mismatch here, and people consider that's an error, a sequencing error, which is a little bit stringent. It's sometimes that they're maybe real, but I think those numbers are really, really tiny, and this type of evaluation is acceptable. So this really tells us sometimes that sequencers reported something, we probably cannot really believe them, and we need to do some recalibration and to correct this. Actually, we'll talk about the recalibration, but the point here is that the blue lines here are the recalibrated ones. So you can see after their algorithm, it can be really come back a little bit. And the question here is, what is causing this miscalibration? So if they reported something 40 as a quality, and actually it's less than that, what can cause that? So they further evaluate a couple of things. And the first thing they evaluate is the machine cycles. And this, we know this is for the Illumina GA2. This is the earlier Illumina platform. It's 35 base pair arrays. Now it's 50 base pair. You can see that Illumina really is lying that all across the board. So regardless of whether it's the beginning of the RAID, which is the first cycle, or the end of the RAID, and all across the board, the accuracy, the reported accuracy is pretty bad. It's really in the negative category. And for the 454, it comes to this type of really bizarre shape. And for solid, so solid is stupid. This is the evidence for that, right? They reported something, like let's say they reported 30. Actually, the real quality is 40. That means they underreported for some of the sequence rates, especially in the beginning of a RAID, but towards the end, they are not very accurate anymore. So it seems that solid probably is not really accurate reporting as well, but they are not doing that intentionally. This is something wrong with their chemistry or things like that. I'm sure that Illumina didn't do that intentionally as well. All right. So for the HiSeq 2000, so you can see this is the pair ends. So you can see this goes to both directions, and this is the beginning of the race and the end of the race, and this is across the board. The reported quality is kind of not as good as they reported. Does that make sense here? All right. And another potential thing that can affect this reported quality is a sequence composition, meaning that for here, they only consider the dinucleotides, so AA, AC, AGAT. We know that some sequences are really harder to sequence comparing to others. For example, CG regions are really hard to sequence for some reason, okay? And so they want to look at whether for different dinucleotides, there's some systematic error here. We can see that for Illumina Genome Analyzer, again, and most of the things are not really accurate. And this is a Roche. So you can see that for most of the platforms, this is CG, and they kind of miscalibrated. Okay? That makes sense? All right. So you can see that this part is, some of them is minus 5, minus 7, 8 category, okay? So what that really means is these higher than empirical quality scores really provide false confidence in these spaces. The machine reported Q25, for example, meaning that the base with quality score is 25, and the p-value is supposed to be 0.003. If you do the calculation, that's pretty good. But after recalibration, it becomes Q20, and that means that the p-value is 0.01. Okay, not as good. So we know that for some of the dinucleotides in some other platforms, it's missed 7 as a unit, and this is even more dramatic than this. So this is using 5 miscalculation as example here. So these are the things that really need to be considered. Yes?
I have a question about the p-value. So does it mean that if the p-value is 0.01 and you are like sequencing 200 nucleotides, that means your expectation of error is 2?
Yes. That's exactly what that means. P-value is by random. If you sequence 200 nucleotides, there's 2 sequencing errors. Okay? And this problem occurs on all major platforms. All these actually have it. And the Illumina being the worst offenders in some conditions, and the quality score is offset by 10, which means the p-value, if they reported, is 40, which is the highest possible reporting. And actually, the p-value is 10 times less than that. So we just need to be careful about this. So what is causing this recalibration? There's many different things. So we mentioned that machine cycles, which the positions in the read or the sequence compositions are possible contributors. And here are really three covariants. The reported quality score, okay, by the sequencer, the position within the reads, this is really the machine cycle. If this is if this is 1, that means the beginning of the reads. 50, that means the end of the reads. And the preceding and the current nucleotides, so this is dinucleotides, if this is AA, AC, or GCG, or things like that. And there's a probability of misalignment, and we can do the calculation. And is there any other covariance? And actually, there's many other covariants as well. So here, what I'm showing here is only for some example. Okay, come back to this. This is for the Illumina HiSeq 2000. So you can see that reported quality and across the board from the very beginning to the end, and it kind of to be over optimistic in the initial reporting, and regardless of the position, and it's kind of too optimistic, and also for most of the nucleotides, it needs to be corrected as well. So how to correct it? That becomes a question. So this covariance and reported quality, machine cycle, and dinucleotides are subsequently applied through a piecewise tabular correction to recalibrate the quality score of arrays in the BAM file. Okay, I was trying to read it. Okay, I got it from their website. All right, I was looking at this for 10 minutes and didn't understand what it really means. Okay, but it's clear that the covariants are this reported quality, machine cycles, and dinucleotides. So let's get something more meaningful. And I stare at this for a while, and probably it's pretty hard to understand as well. Although it's become, for some mathematicians or statisticians, it's become more clear. And I finally got it, I think. So let me explain to you what I understand here. So if this is the reported quality, let's say this is a 40 or things like that, okay? But let's say this is Illumina platform. And the first one is I want to see this, so this is a QR, okay, this is reported quality. And this delta R, which is in the equation, is the error for all the rays with reported quality as R. Meaning that if they reported 30 as a quality, I'm looking at all the reported 30 rays and what is the empirical quality. If it's 25, that means the offset, this delta R should be, this error should be -5. So I'm going to correct based on this R, this reported quality. And next step, I want to look for this reported quality as 30, but the machine cycle, because we know that it's especially for Illumina, for the earlier ones, they were over-optimistic, but for the later ones, they are becoming better. So this is something that we need to correct it as well. So these are the error of our rays with reported quality as R and the machine cycle as C. And so if this is towards the end of the race, I probably want to give it a little more credit, because this part I may overcorrect it. So it comes back a little bit. And next one is I want to see the reported quality together with the dinucleotides. If it's a CG in this region, it's harder, so I'm going to give a penalty here. So this is this red one is a delta RC, and this one is a delta RD. Okay? You probably still don't understand what that is. And this final one will be the recalibrated quality. All right? So this is based on my understanding. Let's have an example. So hopefully this will be more clear. So there's a one base, reported quality is 30. That means the p-value for this is 0.001. That is clear. We already got through it. Okay? And in the experiment, the average empirical quality for all the bases were reported as 30 is actually 25. And the Illumina light here. All right? So this delta RQ is -5. Okay, so this is calculated. And next one is the base locates in the 5th location in the race. The average empirical quality for all the bases with reported quality of 30 and at the 5th location is 27. I'm just making it up. It's 27. But it's better than this 25. So I'm giving it a little bit credit. So delta QRC is 2. This is just because it locates in certain specific location in the reads. So give it a bit more credit. And also, we also know that CG dinucleotide is pretty hard to sequence. And when we look at all the reported quality as 30 and the CG dinucleotide is CG, the empirical quality is actually 23. So this part, the delta is Q is -7. Okay, so because this QC is CG dinucleotide is just too hard to do. And then eventually the recalibrated quality for this part is add up together is become 20. Okay, so start from 30, that is reported quality, and the recalibrated quality is 20. That means the p-value dropped from 0.001 to 0.01, and which is a 10 times a difference. Does that make sense? So this is a so-called stepwise. Okay, you can still, if you're interested, still go back to the initial equations. But I think it's a talk about the same thing, right? Okay, let's look at the performance after the recalibration. So you can see this is now we are staring at this blue dots here, not the purple. Purple ones are the ones that before the recalibration just reported. And after this recalibration, you can see that most of the things that go back to the original place they're supposed to be. Okay, but there's this on this 45-degree line, and for this is accuracy. It becomes 0 here, and here is minus something for the initial one. And for the dinucleotides, everything go back to around the 0 line. All right. So, if some people hand you their data and told you they did the analysis, you want to ask them, did you recalibrate the base quality? And say what that is, and that's a pretty good indication they didn't do that. But you probably want to really understand what that is. And for the informatics students, you probably even need to know how to do it, all right? Because that's something that people expect you to do. Okay. So we are talking about the reported quality to do this recalibration. The reported quality, the machine cycles, and the sequence composition, which is the dinucleotides. And there's many other covariates as well. So including a machine cycle, we are talking about the combination of the base, and which makes sense. Here is the number of consecutive previous bases that match the current base. So whether there is a homopolymer there, okay? And the mapping quality assigned to this is another covariant. And the minimum base quality, position covariant, this position along the length of the read. And for Illumina, this is the same as the machine cycle, which is this one. And for some other weird type of platform, it's still different. And the primer round for this base, and the only meaningful for solid rays, I don't understand what that is. The reported base quality score for this base, and the read group of this rate, meaning the barcode for the barcode samples. And all these things are kind of a different covariance that you can build into this recalibration process. Actually, for the we'll talk about that maybe next time. Maybe I will just send through emails about when we do the homeworks. And in the GATK package, there are pretty established programs to do this, and these are actually the covariants that are included in the GATK package. Okay, if you don't know how to use it, I don't know either. And someone in my lab will know that, which is good. Are we okay with the quality recalibration, the base quality recalibration? These are really something that when you talk to people, did you do that? You say, what's that? That really make you feel good, right? All right, before we go into the variation quality recalibration, which we will do that in the later stage, and I first need to go through the variation identification, Okay, and today is going to be a long lecture. I'm sure that I will have something hanging over. Cannot finish today, but that's okay. Today is the hard part. It's not as exciting as last time, but this is something you really need to know. Okay. So for the variation identification, again, when people hand you their data, they say, "Oh, I already calculated the variance." Okay. Well, you want to step back a little and ask him, what did you do for this? What do you mean by variation identification? Okay, and then you want to think through whether the experiment you are doing is really fitting with the approach they are using. And the first one is for the diploid genome, and this is the most straightforward one. What I mean is, if you got one individual, you sequence their sequence. Okay, so we have a pair of chromosomes, one from mom, one from dad, and that's pretty simple. But for the multiple individuals and for the cancer genome or pool sequencing, they are different. We'll get into that. Yes? Sorry? Not really. It's a sequence of one individual. It can be used for that, but those are most on the mitochondrial DNAs, not really regular DNAs. But here is it's for the disease, study the disease. Okay. But anyways, let's start from the most simple one, which is diploid genome. Okay. So some people may have the question, once you got many reads that cover this one area, why variation causes the issue? It's supposed to be very straightforward. If you see this is supposed to be an A, and now it's a G, and it's a variant, right? So, but let's take a look at an example. So here is, if the reference genome here is everything is the same, but this is A, and when you sequence your samples, there's 11 reads covering these regions, and actually 10 of them show as a G and 1 show as a C. That's a pretty good indication that this C actually is a sequencing error, and this particular location is a homozygous variant. Okay? Does that make sense? Because we have really two chromosomes, a pair of chromosomes for every genomic location that we're supposed to have half of the reads from dad genome, half of reads from mom genome. So if we see something like this, hugely imbalanced, that's more likely to be a homozygous location variant. Does that make sense? And sometimes we see this. Okay? If this is location 5 is G and 6 is A, that is very likely to be a heterozygous variant compared to the reference genome, right? And half of them are from mom, half of them are from dad. I don't know which one is from which, but this type of thing will make sense. So this is a heterozygous variant, homozygous variant. And sequencing error here can potentially cause trouble, but those should be considered. But how to call a variant? So if I got, let's say, 10 reads in this region, if I get 10G, 0A, and I'm very safe this is homozygous, and it's a variant. And if I got a 5G, 6A, okay, for this reads like this, and that is a pretty good indication it's a heterozygous. So we already went through this. But what if this becomes more and more tricky? Okay, if this becomes 9G and 2A, it's still homozygous. Is that right? If it's become 6G, at this I'm pretty confident this is still heterozygous. This is 3G, 8G, 3A, and 7G, 4A. All these thing numbers will show up. So now you're facing what is the cutoff, or where should I draw the line to call this is a heterozygous or homozygous or things like that. So it's become not that straightforward. Okay, you guys are appreciate that? All right, that's good. At least that we are working on something important, right? It's not just you throw the numbers, it comes out. But there's something we can be of help. So there's the factors to be considered here, which is the first one is the number of arrays supporting each genotype, and here is 11 arrays. Sometimes we want over 20 arrays or even 40 arrays. Some experiments we have over 150 arrays coverage. And the base quality for each nucleotide is important. It needs to be considered as well, right? Because if this is a C and the quality is really, really bad, and I probably don't trust it, right? It's just that alignment quality is important. Okay, this is really critical for the alignment quality. If I misaligned, whatever variant I'm calling here doesn't make any sense. Okay, so this is needs to be considered, and sequencing depths, and of course, and sequencing error, and machine-related sequencing error, and these are needs to be considered. And the output after consider all this should be the probability of each genotype. If this is a diploid genome, we have only 3 possibilities for that particular location, right? If it's AA, AG, or GG. So homozygous reference, homozygous alternative, or heterozygous. Is that right? So the output for this is we want to, based on all these cofactors that we observed, we want to see what's the probability for each genotype and which genotype it is. And what we are going to use here is something called the Bayesian approach. And Bayesian inference, again, this is from Wikipedia. Let's go through this. It's a method of statistical inference in which evidence is used to update the uncertainty of a parameter and the prediction of a probability model. Again, this is something that doesn't make sense anyways. So these are something that if you understand, it's straightforward. If you don't understand, I cannot make you understand in a couple of minutes. All right. So this is something. If you're good, you're good. If you're not good, you probably don't care. All right. So let's just go through this and understand as much as possible. So if we have a model and this is the data, right? And if we have the the distribution and what type of statistical model, and I'm pretty good at do the simulation type of thing to see what the type of data I'm going to get, right? But that's not the case. For most of the approaches here is basically I have a model, but I don't know the parameters in the model, okay? However, I do have the data. I want to come back to guess what are the parameters in the model. So those are, am I right for the statisticians? Is that okay? All right, good. So for our case, those are in general Bayesian type of approach, right? But for our case, for variant identification, the model here is actually the genotype. It's AA, AG, or GG, okay? And the data are the reads. So what we are having here is we know the genotype, but we don't know the parameter of the genotype, meaning that which one is more likely. And for this particular case, and the data, we do have the data. So this really fits into this Bayesian type of approach. And for this one, you probably can pretty safe to tell me that, is that this region possible to be a AA? Tell me that. So this 11 rays and then 10 of them are G, 1 is C. Is that possible for this part is a AA? It's possible. It's just unlikely, right? So this is a choice of words. It's possible. Anything is possible. It is very possible. Not very possible. It is possible. This is AA, and every single raise here is an error. Okay, it's just that the possibility is low. So the probability is very, very low. You can still evaluate that. Is that possible to be an AG here? Yeah, it's possible. It's just unlikely, so it's low. But is that possible to be GG? Yes. It's possible and very likely. So basically what we want to do is for each individual genotype, we want to calculate the probability that our data is supporting this. Okay? And for this one, probably you want to go for GG. All right? And the equations, I would say, can be complicated. Actually, you cannot be simpler than this for the statisticians. But for the biologists, it can be complicated, right? So for one locus, and there's an n raised here, and k raised support A, n minus k supports G. That's pretty straightforward. So there's three possible phenotypes, and AA first is AA. If it's AA, and we are observing this, that means we are observing n minus 1 arrows in n rays. Is that right? So if the genotype actually is AA. For the example that we had, it's like we observed the 10 arrows on the same location among 11 rays. And given the sequencing error is only 0.01%, 0.01, it's really, really unlikely we make 10 mistakes out of 11. But still, that's the way to look at this. But if it is AG, that becomes a little bit more complicated. That's a binomial model. In theory, that we should have half A rays and half G rays, right? I mean, in theory, the probability that we observe Ka n minus Kg follows this binomial distribution. We'll have a couple of slides to show you what binomial is, but in the later sections. And if it's a GG here, and actually the probability is showing here. It's observing K arrows because we observe the K rays support A and in N rays. Okay? So all the three genotypes or possible genotypes that we can calculate a probability out of it. And there's another information we need, and for the Bayesian people, that they like to have something like called a prior, okay, which means they make some assumption, which never can be right. They just assume it's right, and then they calculate something they assume it's right. So, which sometimes it's wrong. Okay. But here, the prior of the probability is they have to make an assumption of the heterozygous rate. So the R here is the probability you are observing a heterozygous, which is R, and here is the probability to observe a homozygous, which is 1 minus R divided by 2. And R is a predefined probability of observing a heterozygous. Okay, and this is what Hai mentioned, the SAM2s. When you use the SAM2s to count the variance, and this is exactly the approach that SAM2s is using. Okay, and then they for the known SNP loci, they use R as 0.2. For the unknown loci, they use R as 0.001. Why? I don't know. And it's based on approach, right? So they just assume it's right, and hopefully it's right. But this is the way that they do that. And I'm not going to go through this. So they will calculate something called posterior probabilities, and given the rays that we observe, and calculate the probability of each individual genotypes. Okay, and actually it's pretty straightforward. And then eventually they will calculate the variant quality, which is still this type of Fred scale type of evaluation. Does that make sense? So given all these equations but still coming back to most simple case is you have three genotypes, AA, AG, or GG. Here's all the rays you observed. Okay, and you want to see that given genotype A, what is the probability I observe these rays? Given genotype B, what is the probability? Given genotype C, what is the probability? And then you calculate the probability that which genotype is most likely given the rays you observed. All right? For the statisticians, you don't have to knock your head. I know you got it. But for the biologists, you need to it takes some thinking and to get through this. There's some additional comments that of this approach, especially this. So the method I just told you is very, very simple, but that is the one that is most commonly used. That is the one that included in the SAM2s. And this method only works for diploid genome. Just make sure you know that, okay? If you are working on something called pooled sequencing or RNA sequencing, you don't want to use this base color. And this is a mistake a lot of earlier studies, and at least for the initial analysis they made. And it requires substantial coverage for each genomic loci, and preferably over 20x coverage. And I think for the current technology, this is pretty simple to achieve. And but sometimes this is not always the case. For the exon sequencing project we had, most of the coverages are much more deeper than this, so don't worry about that. But for the whole genome sequencing, especially when we look for the 1,000 genome project, they have only 4x coverage for each genomic, by average genomic loci, and this really, you cannot really use it and to do the base calls. For low to moderate sequencing coverage, this won't work, and this will lead to undercalling heterozygous. Right. And one assumption here is we are having here is also independence among race, which may be violated in the presence of alignment errors or PCR artifacts. Okay, so the general equation I just went through. So it's a still approximation. You cannot really assume they are perfectly correct, but in general it makes a lot of sense. Does that make sense? So what I talk about is that so we observe so many rays, assuming this is a diploid genome, which we have one chromosome from mom, one chromosome from dad. In other words, for any genomic location, they're only supposed to have 2 letters there, right? If it has 3 letters, there's something wrong with our analysis, most likely, right? If you're working around. So, and this is this is a very common approach used in some tools, but it will only work for diploid genome, and also only work for a pretty substantial coverage. Now we're going through a little bit more, which we won't get into the equations too much, but just general conception that for 1,000 genome sequencing, and once they got only this 4x coverage, how you are going to do with it, because this base call approach is not working. It doesn't have enough power to pick up the variations. So this became a very active research area, and of course the people in the 1,000 Genome Consortium, they have to figure that out. And so going back to this single sample, single individual genotype, so these are the prayers of the genotype. So I made the comments that in the Bayesian type of approach, they always need to make a prayer, which is some type of assumption that they hope it's right. And the assumption here was made is the heterozygous rate is R for that particular location, and they even further say for known ones is 0.2, for unknown ones is 0.01. This is not necessarily correct, okay? And especially it can be individual variant based. So R can actually be estimated from a larger population. So think about that for 1,000 genome project. Yes, for each individual we have only 4x coverage, but we have 1,000 individuals. Okay, for a larger population, and this data can really help us to estimate R values. Okay, so not really arbitrarily define one, and this R value can be optimized as well. Of course, it's another a priori, it's another Bayesian type of approach, but it's more accurate. It uses more information and to derive this R. In general, the use of information from multiple individuals when calling genotypes for a single individual should be of great help. Okay, and so here is the general approach. We're not going to go to the equations. Okay, so these are n individuals, and each one has a genotype, and you got allele frequency and the genotype frequency. So you're just basically you use your genotype of frequency, and then to estimate allele frequency and coming back a circle and to refine your parameters. Eventually, you'll make a more meaningful inference. Okay, I'm not going to get into that, and there are actually at least 10 pages of equations. Okay, if you want to go into that, you're welcome. And we put the documentation into the on-course system. All right, but I'm not going to do that because I don't understand that. But the point is, it seems it worked pretty well based on what their conclusion is. And here is one of the conclusions. It's a variant call based on multiple individuals dramatically increased accuracy. This is especially true for low coverage sequencing, such as 1,000 genome. By average, it's only 4x coverage. So you can see this is the blue ones are based on the variant call based on single samples. It's not bring multiple individuals together. It's based on single samples. And the red one is based on multiple samples. So you can see it start to improve the performance pretty well. And the black one, actually, there's another piece of information I didn't mention, which is the population-based parameter tuning. And once they integrate the population, because we know the population of these 1,000 genome samples, and then you can see the accuracy really are improved dramatically. So we can pretty fair to say that although the 1,000 genome project, the coverage is pretty low, but given the depth of analysis we are having, the conclusion from the 1,000 genome sequencing is very credible at this stage. And if you're interested in this, and feel free to go to these reviews in the Nature Genetics. Okay, so for the second second part of the multiple individuals and for the variation costs, and this is just I'm just giving you the concept. So we are not going into the details. But the point here is using a large amount of samples can help us improve your yield for individual samples as well. So everything I'm talking about, including the realignment, including the quality recalibration, and including everything I'm going to talk about today, I'm just giving you a hard time to get into the mathematical equations. And the point here is I really want you to appreciate the assumptions that are being used here, not necessarily go into the equations. And once you really do it, there's no reason to do any programming on this. They have already very well packaged the software, so you can really type in a few Linux commands and wait a few days, and most of this analysis will be done. So it's not that hard, but their principle need some time. We need some time to figure it out. Okay? Any questions here? So are we all okay with the variant call? So these are the statistical series I want to introduce about the variant call part. Okay? Are you all okay? Or are you all confused? All right. But for the somatic and the pooled sequencing, actually this is not a diploid genome. So everything I talked about in the previous ones, they are diploid genome. We have a pair of chromosomes. But for the cancer genomes, only a small proportion of the cells, they probably will mutate. Okay? Not all the cells have that. And for pooled sequencing, depending on the number of samples in the pool, only a fraction of variants are different from others. Okay? If we have 100 individuals, their DNA sequence put together, maybe only 1 or 2% have that variant, and that is a very small percentage. So this is really violated that 50% the diploid genome assumptions. And everything that we were talking about this binomial distribution and things like, and prayer and things like that will not hold anymore. For the RNA sequencing, allele imbalance. So we can have one allele, mother one express a lot, father one doesn't express a lot. So it's not 50-50 anymore. And that can cause trouble using the approach that we just introduced. And so for this one, so remember when we initially listed this in the example, this is a homozygous variant, right? So that is our conclusion. But now, the same data will have different interpretations. For diploid genome, this is homozygous A is a sequencing error. And for the somatic sequencing, and for the cancer cells, for example, this may represent a novel mutation that is only presenting in a few percentage of cells. Does that make sense? Okay. And for the RNA pooled sequencing, this is a rare variance. If I have 100 individuals pulled together, if I observe this, of course the depth should be much higher than this. And then this is represent a rare variant. Yes?
So for something like a somatic, how can you differentiate between a sequencing error and a...
We'll get into that. That's a good question. How do I know this is sequencing error or a somatic variation? We'll get into that. And for RNA sequencing, and the G is allele, so the interpretation here is G is allele, it expresses much more than A allele. That can be the interpretation, right? So for the exactly same patterns we are observing here, we are really drawing different conclusions, which is always not a good thing. And diploids, but chromosomes are not imbalanced for this type of situations. The key is what you just exactly mentioned. Separate the minor allele from the sequencing error. How do I know this A is a real minor allele or it's a sequencing error? And the assumption is 2 possible nucleotides. Still, it's a diploid genome. Okay, for one location, it can be A or can be G or can be a pair of things, but better not bring in the third letter in it. If there is, the analysis itself may need to be changed a little bit as well, which is possible, right? We know that for a very small percentage of genomic loci, there's a trial alleles, right? So there's one location can have 3 different letters, not in one individual, but in our human population. But when we put the samples together, it could happen, right? And for the cancer cells, for the mutations, how can you control this mutation in this way or that way, right? So it's possible, but just not what we are trying to figure out. So assumption here is still 2 possible nucleotides. The third most abundant one is an error. If you think it's not a reasonable assumption, and we need to design some other approach to fix this. But this is our assumption. For example, for one genomic location, if we got 100A, you have 10G or 2T, and you probably will consider A is a major allele, G is a minor allele, T is an error. Is that fair? Okay, this is not statistical yet, so we will get into that. But based on this data, if you just stare at this data, this is probably the conclusions you can make if this is RNA-seq or a pooled sequencing. Does that make sense? But everything that we talk about the binomial distribution thing, that won't work. But is this correct? Is that significant significance? So there's several published approaches and to study this. And the first one is published in Nature Methods, 2009, called SNP-Seeker. It's only a 2-page article, and the entire algorithm is in the supplementary. So it wasn't even in the main text. And they were based on large deviation theories. I'm not going to go into that. I got these slides done last night. I didn't have a chance to really read the several pages of equations in there. And another one is called SNV Mix2. It's a probabilistic binomial mixture model. Okay, it can be really, really complicated as well. And this is published in Bioinformatics in 2010. But there were 2 papers using this approach, one published in Nature, the other published in New England Journal of Medicine. And both of them are trying to figure out the somatic variations, okay, in cancer cells. So I assume this is very credible, but I did use it a little bit. Maybe I didn't use it right, but result really, I don't like it. But so probably It's probably because I didn't use it right. But I don't want to give additional comments on that, and this is another possible solutions. And the third one is called SNVR. It's a binomial model. It's published in NAR this year. And basically, binomial means they have two binomial models stacking together. One is the number of individuals carrying minor allele follows a binomial distribution. So there's so many individuals. How many individuals carry it follow binomial? But within one individual, the number of rays follow a binomial distribution as well. So there's two levels of binomial models, and they did something of that. Assume that the calculation should be very quick. I haven't tried it, so to be honest. This just comes out not long ago. Okay, and a couple of things that I'm trying to get is actually Yang work, and we designed our method is called based on a beta distribution. It becomes more complicated, but let's try to go through this. So we keep talking about the binomial distribution. For the statisticians in the room, you can sleep now, okay? But for everybody else, so what is really the binomial distribution? So the real good example for this is really toss the coins. So you throw the dimes up, and it's face or head, and things like that. And if it's good coin, and probably we are getting probability of 50% head, 50% tail. And the number of heads in 10 tosses follows a binomial distribution. So if I throw this coin for 10 times, how many times I'm going to get a head? 5? Yeah, 5 will be my guess as well. But if I bet money on it, that's what I'm going to. But there's a probability there no head, right? You just got not really lucky. You just throw 10 times, everything is a tail. Is that possible? Yes. Is it likely? Not really, right? Come to the same problem. So, and the probability for this is really, really low. The same thing happens on the reverse side. So all the 10 tosses and becomes a tail, all head, and the probability is low. So we know that the largest probability happens at 5, right? If I bet money on that, I will probably bet 5. And actually it follows this type of distribution called a binomial distribution, and it's 0.05. And actually, based on this equation, you can calculate whether this is, if it's 6 times or 4 times, you still got 21% and things like that. You can always make your calculation. Does that add up with 100%? I may make some mistake. Yeah, but anyway, so that's the point. The point is that this is a binomial distribution. Well, but why does the coin tossing thing will help us, right? Let's just go back to this. For the n is, if this is n is the number of times that we throw the coin, and the p is the probability of a head for each tossing. If it's good coin, that will be 50%. And n is the number of head after 10 tosses. That's what the equation means. But for the variation identification for our problem, n here is the number of times we throw the coin, and here is the number of sequence reads that cover that particular loci, right? If I got one particular genomic locus, how many reads cover that? Okay, and actually, total number of reads will be the equivalent to the number of times I throw the coins. And P here is the allele frequency for the pooled sequencing, right? So if it's not 50% anymore, right? If it's only 10% of populations have this variation, and this is P will be 0.1, things like that. If 90% have it, that will be 0.9. And the n here is the number of rays representing one allele among the total number of n rays. So if you think it through, and this is exactly the same type of problem, right? That's why binomial distribution is helpful. But what happens with there's beta distribution? What that is, right? So go back to our question here. Is the P is the allele frequency, n is the total number of rays, and this small n is the number of rays supporting the minor allele. Okay, if allele frequency and n and the total number of reads are known, and this small n will follow a binomial distribution. We went through it. However, for our case, if large n and small n are known, so the total number of reads and the number of reads that supporting minor allele, and this probability, the allele frequency actually follows a beta distribution. Is that right? The statisticians in the room? You probably want to go back to check your books. I hope it's right. All right? And so this really follows a beta distribution. So this is some type of exact approach that we are trying to use, and let's see what the data really look like. And this is not really readable from the back. Okay, so for the red ones, this is okay. I'm reading this. It's closer to me. So alternative is reference. You got 18,000. The reference is 2,000. This is for the red one. And so allele frequency will be 10%, okay? Total number of reads here is 20,000, and 2,000 I'm seeing alternative allele. So it's a 10%. And the second is one is 10 times lower. So total number of reads is 2,000, but I'm seeing 200 of alternative, 1,800 reference. And the last one is really not very deep. I think I have 200 reads cover this location, and 20 show the alternative, 180 show the reference. But all of these numbers, if you think it through, that implies that the allele frequency is 10%, okay? But with the deeper sequence, we are having more confidence that we are doing right. So this is what the distribution look like for the deeper sequencing. So you can see the band is really, really narrow. We have, if we define the 95% confidence interval, and we are really fine-tuned to a very small range of allele frequency. Well, for this particular one, since we are not sequencing that deep, so the distribution will become much fatter. Okay, while we look for the total number of the confidence interval, and this interval will become really, really big, right? So that means I'm thinking from anywhere from 5% to 50%. That's not really meaningful. If you say, oh, I got this, I think it's from 9% to 11%. That is much meaningful. So we want that confidence interval to be really, really as small as possible. And with deeper sequencing, it's clearly that that can help us to narrow our confidence interval. Still make sense? Okay. Now comes to our seeking for the differences between minor allele frequency and the background noise. It comes back to the original question. If there's a minor allele here, how am I convinced that this minor allele represents the real thing, not the sequencing error? So what our approach here is we will compare the second largest number versus the third largest number. Okay, if for one location there's four numbers, how many A, how many G, how many C, how many T? Okay, and the second largest, most likely, it can be nothing, can be sequencing error, it can be a minor allele. For the third largest number, it certainly is a sequencing error, okay? And because we are still on the diploid genome, and our hypothesis testing will fall into this. So for the alternative one, that will be the second largest number is is significantly higher than third largest, which is represent the noise. And then we will be able to calculate the p-value. So you can imagine there's a peak representing the sequencing error, there's a peak representing the alternative allele, and how well they distinguish each other. If they doing things like this, overlapping with each other, I don't have enough confidence to say this is not a sequencing error. But if the sequence, the depths become much higher and the peaks becomes narrower, their overlapping becomes less and less, and the further away they are, the more possible that they are minor alleles, not sequencing errors. Is that answer your question? See, I'm very serious. Use 10 slides to answer your question. And is that forward? Okay. So the summary for the variant identification is methods of identifying variants from sequencing data really varies. And it depends on the question we have, whether it's a diploid genome for whole genome and exon sequencing. We use a very early approach that we talk about. And for the low coverage, we use multiple individuals. For the unbalanced data, we can use one of the approaches I listed there. And of course, ours is one of it as well. Okay? We'll take another break.